Hi guys, in this video we are going to discuss about one of the important topics in molecular biology, the DNA repair mechanisms. So these repair mechanisms are very important not only from exam point perspectives. Suppose let us imagine that these repair mechanisms are not there. What will happen? The errors in the DNA cannot be repaired. It will remain as a permanent error, which we call it as mutation. So these repair mechanisms act as a defense mechanism and reduces the rate of mutation and the prevalence of cancer and other hereditary diseases. Okay, so that much important is our topic. Damage to DNA can happen during two instances. First of all, during replication process, and the second is outside replication. That means when the cell remains in the other phases of cell cycle, mainly in the G0 phase or resting phase. First, coming to the replication part, during replication, due to the proofreading activity of DNA polymerase, the chance of error is very less. Okay, we can say the rate of error is just 1 in 10 to the power of 6 bases added. Even such a rarest error is not permitted by our body as it can lead to mutation. Okay, so such errors are called as mismatch errors. Normally in the DNA, adenine will be paired with thymidine and guanine with cytidine. Okay, so A to E and G to C. Okay, so this is called as base pair rule, but here it will be mismatched. Okay, so to repair such type of errors, we have a specific type of repair mechanism called as mismatch repair mechanism. Okay, now coming to the extra replication part, when the cells are in the resting phase, the DNA within it is exposed to a lot of physical, chemical and biological assaults caused due to metabolic activities, the chemicals we take in the form of food preservatives, food additives, even the medicines we take, the ultraviolet radiations, in X-ray radiations, and even the smoking, okay, or the chemicals present in the smoking. So all these agents can damage DNA and it can produce three major types of errors. First one is single base error, where only one base in a strand will be damaged. Next is multiple base error, where more than one, two or more bases of same strand will be affected. Okay, but if the two bases in both the strands are affected, it leads to double strand breaks or sometimes cross linkages. Okay, so to correct these different types of errors, we are equipped with different and specific type of repair mechanisms. For correcting single base errors, we have base excision repair. For correcting multiple base errors, we have nucleotide excision repair. And for correcting double strand breaks and cross linkages, we have non-homologous end joining and homologous recombination. Okay, so totally there are 5 major types of repair mechanisms present in our body. Okay, so to start with, mismatch repair, base excision repair, nucleotide excision repair, non-homologous end joining, and homologous recombination. Got it? So this is just an overview about the DNA repair mechanisms. Okay, so now we will see in detail about each one of these things. Okay, so first let us start with the different types and causes of DNA damages. Okay, so in that we have already seen there are single base errors, multiple base errors, and double strand breaks. Okay, so in that the major single base alterations are depurination, deamination, alkylation, and sometimes oxidation of the individual bases. So out of these different types, the most common single base error is deamination of cytosine. Okay, so this deamination will be caused either spontaneously or sometimes due to the chemicals like nitrites, nitrous acid, and bisulfates, which are mainly present in the food preservatives. Okay, so when the amino group is removed from cytosine, it gets transformed to uracil. It gets transformed to uracil. If this transformation happens, it leads to mutation and related diseases. Got it? Yes. So to prevent this mutation and to correct this error, we are equipped with a repair mechanism called as base excision repair. Okay, next coming to two base alterations. The most common one is pyrimidine dimer or thymine dimer caused by ultraviolet radiation. To correct this error and to prevent this error, to prevent this mutation caused by this error, we are equipped with nucleotide excision repair. Okay, next coming to chain breaks and cross linkages, they are mainly caused by ionizing radiations like X-rays, UV rays, X-rays, gamma rays, etc. and also by the free radicals produced in our body as a result of metabolism. Okay, so to correct these errors, we have two different types of mechanisms. One is non-homologous end joining and the other one is homologous recombination. Okay, so that's it about the different types and causes of DNA damages. Now we will see in detail about each one of these repair mechanism. To start with, we'll start with mismatch repair mechanism. About each one of these repair mechanism, you should know three things. The first one is what type of error it repairs. Second one, how it repairs, and finally, what will happen if it is not repaired. Okay, so I'll be discussing all these repair mechanisms only mainly under these three headings. Okay, so first, what type of error it repairs? We all know that mismatch repair is exclusively for the mismatch error caused due to defective replication or due to the defective action of DNA polymerase. Okay, so let us consider this is the newly replicated DNA which is having a mismatch error where adenine is base paired with cytosine. Okay, so normally adenine should be paired with thymidine or cytosine means it should be paired with guanine. But here adenine and cytosine are paired, which we call it as a mismatch. Okay, so now we will see how this mismatch is corrected and repaired. Okay, so to correct and repair this, we have a group of three proteins which are named as MUT-S, MUT-L and MUT-H. Out of these three proteins, MUT-S is the one which identifies the error. Okay, after identifying, it helps the entire complex to bind to the DNA at the point of error. Okay, so the MUT-S along with MUT-L and MUT-H will identify the mismatch error and it will go and bind at the site of the error. After binding to the error, it should find out which is the wrong base, whether adenine is wrongly matched with cytosine or cytosine is wrongly matched with adenine. Both the possibilities can happen. Okay, so how to find out that means we all know that DNA replication is a semi-conservative process, which means one strand is a parent strand, old strand, whereas the other strand is the newly synthesized strand. Okay, so if at all any error, it should be only in the newly synthesized strand. Okay, so next we have to find out which is the parent strand and which is the newly synthesized strand. Okay, so usually after replication, some of the bases in the newly synthesized strand will undergo methylation, which we call it as post-replicational modification. Usually these modifications will happen after a small delay after the replication. There will always be a lag between replication and this post-replication modifications. Okay, and it is only during that time gap our repair mechanism will work. Okay, so during this time gap, one strand, the parent strand, the old strand will be having methyl group, whereas the new strand, the methyl group is yet to be added. So there won't be any methyl group in the new strand. Okay, so this is used as a marker to distinguish parent strand and the new strand. Okay, so what will happen means our MUT-S, MUT-L and MUT-H protein, they will scan for the methyl groups on either side of the mismatch error. Okay, so once it finds the methyl group on any strand, it will recognize it as on any end. If it finds a methyl group, it will recognize it as a parent strand and it will put a small nick on the opposite strand, which is the error containing strand. Okay, you can see here. So since methyl group is found here, this will be finalized as a parent strand. Okay, so the opposite strand is a new strand which will be containing the erroneous base. Okay, so after distinguishing the old strand and the new strand, the MUT-H protein will make a small nick in the new strand. Okay, now from this open end of the nick, the nucleotides will be removed one by one by an enzyme called as exonuclease till the error base. So from here, the nucleotides will be removed one by one till this point, creating a gap by the enzyme called as exonuclease. Okay, next, this gap will be refilled by DNA polymerase and DNA ligase, thereby completing the process and completing the repairing work. Got it? I will just repeat once again. It actually involves 4 steps. Okay, so the first step is identification of the error by MUT-S protein. Okay, so after identifying, the whole protein complex will scan for the methyl group to identify which is the old strand and which is the new strand. Okay, so after identifying, the mut H protein will make a nick in the new strand. Okay, so from that nick, from the open end of the nick, the nucleotides will be removed by the nucleotides along with the erroneous base will be removed by exonuclease enzyme. Next, the gap will be finally filled by DNA polymerase and DNA ligase. Okay, so this is how mismatch errors are repaired in prokaryotes. Okay, whatever I described so far is happening in the prokaryotes. Okay, in eukaryotes also it follows the same step. The only difference is instead of mut S, mut L, and mut H, we have a different set of proteins which are called as mut S homolog, mut L homolog, and mut H homolog. Okay, so when these proteins are defective, this mismatch repair cannot be done. Okay, so that errors will lead to mutation and leads to a condition called as Lynch syndrome or hereditary non-polyposis colon cancer. Okay, so the defective mismatch repair mechanism can lead to a condition called as Lynch syndrome or hereditary non-polyposis colon cancer. Okay, got it? So that's it about mismatch repair mechanism. Next, we will move on to the second type, base excision repair. Okay, so here also again the same three headings. So first, what type of error it repairs? Okay, so we all know that it mainly repairs the single base errors. Okay, we have seen many different types of single base errors, but out of that, the most commonest one is deamination of cytosine to form uracil. Okay, so how this uracil is removed and replaced by cytosine, we will be seeing now. Okay, so normally in DNA, there should not be any uracil residue. Okay, if it is there means it is an error. Okay, so this uracil formed by deamination of cytosine will be identified by an enzyme called as uracil DNA glycosylase. Okay, so this uracil DNA glycosylase will identify the abnormal uracil nucleotide in the DNA. Okay, after identifying, as the name says, glycosylase, it will break the glycosidic bond between the uracil residue and its sugar molecule. Okay, usually the base and the sugar residues will be connected by glycosidic bond. Okay, so that bond will be broken. So once that bond is broken, the uracil residue alone will fall down, creating a empty site, which is otherwise called as AP site. Okay, AP means absence of purine or absence of pyrimidine, apurine or apyrimidine site. Okay, so here base alone is not there, but the backbone for phosphate and sugar molecule is still intact. Okay, so now this AP site will be identified by another enzyme called as AP endonuclease. Okay, so this AP endonuclease will come and remove the phosphate and sugar backbone along with few other nucleotides to create a gap. Now, as usual, this gap will be refilled by DNA polymerase and DNA ligase and completes the process. Okay, so base excision repair is very simple. Okay, it is almost similar to mismatch repair only. The steps are common only. Okay, so both the repair mechanisms involves 3 steps. The first step is identification of the error and making a small nick. Okay, second thing is removal of the error. Third is replacement of the error. Okay, so first one, identifying and making a nick. Okay, in mismatch repair, this identification and making a nick in the error containing strand is done by mut S, mut L, and mut H protein. Whereas here it is done by uracil DNA glycosylase. Identification of error and it makes a nick in the glycosidic bond. Okay, next, removal of the error. So removal is done by a exonuclease in mismatch repair, whereas by AP endonuclease in base excision repair. Okay, then third step is replacement of the nucleotides. Okay, so that was done in both the condition by DNA polymerase and DNA ligase. Got it? If this base excision repair is defective, it can lead to mutation and lot of cancers. Okay, so there are various lung cancer and breast cancer that have been reported where there is defective base excision repair. Okay, so that's it about base excision repair. Okay, so next we will move on to the third type, nucleotide excision repair. Okay, so base excision repair is for single base errors, whereas nucleotide excision repair is for multiple base errors. That means more than one bases are involved. Okay, so the most common multiple base error is pyrimidine dimer and benzopyrene adducts. Okay, so out of this, I will take pyrimidine dimer as an example, and I will explain you how the process is happening. Okay, so you can see this arrow mark. No, so this is the pyrimidine dimer, which is caused by ultraviolet radiations. Okay, so now we will see how this is corrected. Okay, so here also it follows the same 3 steps. Okay, so first step is identification and making a nick. So here this identification and making a nick is done by a protein complex called as UVR ABC exonuclease. UVR, that means UV radiation. The protein which corrects the error caused by UV radiation is named as UVR ABC exonuclease. Okay, it is not exonuclease, it is exonuclease. Okay, so this is made up of 3 proteins: UVRA protein, UVRB protein, and UVRC protein. Okay, so out of these 3, UVRA is the one which identifies the pyrimidine dimer and helps the protein complex to bind to the DNA at the point of error. Okay, so after binding, the other 2 proteins, UVRB and UVRC, they will make 2 nicks, one on either side of the pyrimidine dimer. You can see one nick on 5' end and one nick on 3' end of the error. Okay, so usually UVRB will make a nick 5 nucleotides away from the error in the 3' direction, whereas UVRC will make a nick 8 nucleotides away from the error in the 5' direction. Okay, so like this, 2 nicks will be made. Okay, so after this, next what will happen means the enzyme helicase will come into action. Okay, what is the function of helicase? It will break the hydrogen bonds. Okay, so it will break this hydrogen bond between these 2 nicks. Okay, so between these 2 nicks, the nucleotides, including the error bases, are connected to the other strand by the hydrogen bonds. Okay, so these hydrogen bonds will be broken by our helicase. So once the hydrogen bonds are broken, what will happen? The lower part, which contains the pyrimidine dimer, will just fall down, creating a space. So once the space is created, as usual, our DNA polymerase and DNA ligase will come and replace it, completing the process. Okay, so this also involves same 3 steps. First one is identifying the error and making a nick. This is done by a group of protein called as UVR ABC exonuclease. Okay, after making the nick, the error strand is removed by the enzyme helicase. Then it is replaced by DNA polymerase and DNA ligase. Okay, so in this, the main enzyme which plays a major role in nucleotide excision repair is UVR ABC exonuclease. Okay, so this ABC exonuclease is in prokaryotes. Okay, but in eukaryotes, the same job is done by another group of proteins which are named as XPA to G as well as CSA and CSB. Okay, so why they are named as XP and CS means when these proteins are not there, it leads to a condition called as xeroderma pigmentosa. That's why XP, and Cockayne syndrome. That's why CS. Okay, so apart from these 2 diseases, we can also find this defective nucleotide excision repair in another condition called as ataxia telangiectasia. Okay, so these are the 3 disease disorders caused due to nucleotide excision repair defect. Okay, got it? So that's it about nucleotide excision repair. Okay, so next we'll move on to the double strand breaks. How the double strand breaks are corrected. Okay, so actually these double strand breaks are slightly unique from the other types of DNA damages. In other types, from starting, even the replication error or single base error or multiple base errors. Okay, in all these damages, the nucleotides on only one strand is affected. Okay, so with the information on the other strand, the replacement can be easily done. But here, what is happening? Both the strands are, the nucleotides of both the strands are damaged, leading to double strand breaks. Okay, so how these double strand breaks are repaired, we will see now. Okay, as I already mentioned in the initial slide, we have 2 different types of mechanism to correct such errors. Okay, so one is homologous recombination and the other one is non-homologous end join. Okay, so first we will see about homologous recombination. Okay, so when the error is detected or when the error happens during or after S phase, it will be corrected by homologous recombination. Okay, so when the double strand break happens during or after S phase, it will be corrected by homologous recombination. Okay, so after S phase, okay, after S phase, how many sets of DNA will be there in the nucleus? two sets. Okay, so two identical or two homologous sets will be there. Okay, so the error in one strand, we can get the information from the other DNA, other homologous DNA, right? Yes or no? Okay, so that's what happens in this homologous recombination. Okay, so what will happen means the double strand break, one end of the break, okay, they will invade into the other DNA synthesized, okay, other homologous DNA, okay, to form an intermediate called as holiday intermediate. Next, the other strand will be used as a parent strand, template strand, and the error will be corrected, leading to completion of the process. Okay, this is called as homologous recombination. Okay, so actually this involves a lot of proteins which needed for this type of homologous recombination, but at undergraduate level, if you know just an idea, overall idea, that much is enough. But what you should know is when this is defective, it can lead to disorders like Bloom syndrome, Werner's syndrome, and it can also increase the susceptibility of breast cancer. Okay, so that's it about homologous recombination. Okay, so this homologous recombination will happen when the double strand break happens after S phase. Okay, but suppose if it happens before S phase, that means in the G0 or G1 phase, it will be corrected by non-homologous end join. Okay, so as the name says, what will happen means here in G0 and G1 phase, we won't have the another DNA which can be used as a template to correct the repair. So here what will happen means the two ends will be abruptly joined, okay, to correct the error. Okay, so once it is, if it is not joined, what will happen? The DNA synthesis cannot proceed further. So it is abruptly joined so that even at the loss of few nucleotides, we are maintaining the replication process. Okay, so this non-homologous end joining is mainly catalyzed by two major proteins. One is KU protein and the other one is DNA dependent protein kinase. Okay, so since we are adding abruptly, there are chances that we may miss few nucleotides in between. Okay, so always this type of mechanism is an error prone repair mechanism. Though it is error prone, it is actually the most commonly used repair mechanism for correcting the double strand break repairs. Got it? But if this repair mechanism is defective, it can lead to a condition called as severe combined immunodeficiency. Okay, so that's it about non-homologous end joining. And with this, I'm completing all the repair mechanisms. Okay, so to summarize, these are the major types of DNA damages and their respective repair mechanisms. For replication error, we have mismatch repair mechanism. For single base error, we have base excision repair. For pyrimidine dimers caused by ultraviolet radiation, we have nucleotide excision repair. And for double strand breaks, we have homologous recombination and non-homologous end join. Okay, so most of these repair mechanisms, they usually follow three common steps. So what are those three steps? The first step is identification of the error and making a small nick in the error containing strand. Second step is removal of the error. Third step is replacement. Okay, so in mismatch repair, the identification and making a nick is done by a group of protein called as MUT-S, MUT-H, and MUT-L. Whereas in base excision repair, it is done by uracil DNA glycosylase. Whereas in nucleotide excision repair, it is done by UBR ABC XZ nuclease. Okay, coming to the removal part, in mismatch repair, the removal is done by exonuclease. In base excision repair, it is done by AP endonuclease. Whereas in nucleotide excision repair, it is done by the enzyme helicase. Okay, and finally coming to the replacement, that is a common enzyme, DNA polymerase and ligase, which replaces and refills the gap in all the three types of repair mechanisms. Okay, so these are the various mechanisms. Okay, so next, finally, what you should know is the diseases caused due to various defect in the various repair mechanisms. Okay, so defect in mismatch repair can lead to Lynch syndrome. Defective base excision repair can lead to breast cancer and lung cancers. Defective nucleotide excision repair will cause xeroderma pigmentosa, cocaine syndrome, and ataxia telangiectasia. And finally, defects in double strand break repairs can lead to severe combined immunodeficiency, Bloom syndrome, Werner's syndrome, and also increases the susceptibility of various breast cancers. Okay, so that's it about DNA repair mechanism. Okay, so if you have any doubt, you can please comment in that. And if you want any other videos, any other topics, small topics, if you want me to give it as a video, please comment in the comment section. so I can proceed, I can come up with the new videos. Okay, thank you.